Good morning all. Today we are going to discuss last portion of the sterilization, that is gaseous sterilization. And this topic in today's discussion include gaseous sterilization, different methods coming under gaseous sterilization, then plasma sterilization, then finally physiochemical method of sterilization. So the sterilizing gases are typically used when exposure to other methods, for example heat or radiation, could damage the materials or equipments. The most common gases used for the sterilization include ethylene oxide, formaldehyde, mixed oxides of nitrogen, then finally ozone. According to the USP, ethylene oxide's ability to penetrate through polymers, cellulosics and other materials allows it to be used for the terminal sterilization of medical devices in their final packaging. So, this gaseous sterilization, it involves the process of exposing equipments or devices to different gases. How? In a closed, heated or pressurized chamber. So this gaseous sterilization, it is more effective technique because the gases can pass through the tiny orifices of the exposing equipments, and this may provide more effective results. Besides, the gases are commonly used along with heat treatment, which also facilitates the functioning of the gases. However, there is an issue. What is the issue? That is the release of some toxic gases during the process. So, it should be removed regularly from the system. So, the mechanism of action for each gases are different. Some of the common gases which are used in gaseous sterilization are given. First is ethylene oxide. It is a powerful alkylating agent which is capable of destroying microorganisms by chemical reaction. And the main reaction is primarily it interferes with the cellular DNA. The destructive mechanism largely follows first order kinetics, and the destruction depends upon the concentration, humidity, temperature, etc. This ethylene oxide nature is, it is a colorless gas and that is flammable and explosive too. The four essential parameters, that is operational ranges of this ethylene oxide is, it should be maintained at a gas concentration between 450 to 1200 milligram per liter, and the temperature should be at a range 37 to 63 degrees Celsius. Then coming is relative humidity. It should be 40 to 80%. That means water molecules carrying ethylene oxide to reactive sites. Now the exposure time, it should be 1 to 6 hours. Now what is the operation? The basic ethylene oxide sterilization cycle, it consists of 5 stages. That means first is preconditioning and humidification, gas introduction, then exposure of that equipments to that gas, then evacuation of the gas, then finally air washes. And this all procedure takes place 2.5 hours excluding aeration time. Now the mechanical aeration for 8 to 12 hours at 50 to 60 degrees Celsius, it allows desorption of toxic ethylene oxide residuals contained in exposed absorbent materials. Now next is the mode of action. The microbicidal activity of this ethylene oxide, it is considered to be as a result of alkylation of protein, DNA and RNA. Alkylation means replacement of hydrogen atom with an alkyl group, and this may result it prevent normal cellular metabolism and also this normal replication. And the mechanism of action is assumed to be alkylation of sulfhydryl group or amino group or hydroxyl group and this carboxyl groups on proteins, and also the imino groups of nucleic acids are disrupted. Now it inactivates all the microorganisms, although bacterial spores too. But there are certain bacterial spores such as Bacillus atrophius. Their spores are resistant than other microorganisms. So for this reason, the Bacillus atrophius, it is recommended as biological indicator in the case of ethylene oxide sterilization. And this ethylene oxide kills viruses and fungi, including yeast and molds, and is compatible with almost all materials, even when repeatedly applied. Now the uses. It is used in healthcare facilities to sterilize what? Critical items and sometimes semi-critical items too, that are moisture or heat sensitive, and they cannot be sterilized by steam sterilization. For the sterilization of heat sensitive items such as disposable plastic petri dishes and syringes. So the heat sensitive items which are sterilized using this method are disposable plastic petri dishes, syringes, heart-lung machine, sutures, catheters, respirators, and dental equipments. Now the disadvantages. What are the disadvantages? It has a lengthy cycle time. First one, then high cost. And also it may cause potential hazards to patients and staff. That means acute exposure to ethylene oxide, it may result in irritation to skin, eyes, gastrointestinal or respiratory tracts, and central nervous system depression. The chronic inhalation has been linked to the formation of cataracts, cognitive impairment, neurologic dysfunction, and disabling polyneuropathies. Next is about formaldehyde gas. Formaldehyde is another important highly reactive gas which is used for sterilization. This gas is obtained how? By heating formalin to a temperature of 70 to 80 degrees Celsius. It possesses a broad spectrum of biocidal activity, and it has found application in the sterilization of reusable surgical instruments, specific medical diagnostic and electrical equipments, and surface sterilization of powders. Formaldehyde doesn't have the same penetrating power of ethylene oxide, but it works on same principle. That means it causes the modification of proteins and nucleic acids. Because of this low penetrating power, it is often limited to paper and cotton fabrics. Formaldehyde can generally be detected. How? By the smell at a concentration lower than those permitted in the atmosphere, and thus can be detected during leakage or other such accidents. Next is about another important gas, that is beta-propionolactone, BPL. It is a condensation product of ketane and formaldehyde. This BPL is a liquid which exerts a strong bactericidal action on the surface of the objects which are immersed in aqueous solution of it or exposed to vapor phase. In the present experiment, it is used in 3 different ways. How? It is being vaporized by heat in air at atmospheric pressure, or vaporized at room temperature under reduced pressure, or it can be applied as an aerosol by means of nebulizers. So these are the 3 methods in which they are applied. It is more efficient in fumigation purpose than formaldehyde and has rapid biocidal action, but unfortunately it also has a carcinogenic activity and a poor penetrating power. And the concentration used for the sterilization of biological material is 0.2%. And it is capable of killing all microorganisms and also very active against viruses. And also it is an alkylating agent and acts through how? Alkylation of carboxyl and hydroxyl groups. Next is about nitrogen dioxide. The nitrogen dioxide is a rapid and effective sterilant that can be used for the removal of common bacteria, fungi, and even spores. Nitrogen dioxide has a low boiling point which allows a high vapor pressure at standard temperature. This property of nitrogen dioxide It enables the use of gas at standard temperature and pressure. The biocidal action of this gas involves what? Degradation of DNA by nitration of backbone, and it may result in lethal effects on the exposed organism because they absorb nitrogen dioxide. Advantage of this method is that no condensation of gas occurs on the surface of the device. Why? Because of the low level of gas used and high vapor pressure. And this avoids the need of direct aeration after the process of sterilization. So there is no need of direct aeration. Another important gas is ozone. It is highly reactive industrial gas, commonly used to sterilize air, water, and as a disinfectant for surface. It has a potent oxidizing property. Yesterday we have discussed about the oxidizing agents. We have told a line about ozone. And it is capable of destroying wide range of organisms including prions without the use of hazardous chemicals, as ozone is usually generated from medical grade oxygen. High reactivity. The ozone is highly reactive, and this may allow the waste ozone, removal of waste ozone by converting the ozone O3 into oxygen O2 by passing it through a simple catalyst. Because ozone is unstable and it is highly reactive gas, so it has to be produced on site, which limits the use of ozone in different settings. That is the disadvantage. It is also very hazardous and thus only be used at a concentration of 5 parts per million, which is 160 times less than that of ethylene oxide. Next is about plasma sterilization. This is a recently introduced sterilization procedure, that is STERRAD and Plasslide, and it is used for creating a plasma state. How? So as to maintain a uniform vacuum inside the chamber. Vacuum means without air. The chemical sterilants which are used in this type of sterilization are hydrogen peroxide alone or a mixture of hydrogen peroxide and peracetic acid. Active agent is UV photons and radicals. Example, oxygen and hydroxy. This may kill the microorganisms and spores. Low temperature is maintained less than 50 degrees Celsius. So it is best for heat labile surgical instruments. Finally, last one is physiochemical method. So we have discussed physical methods, chemical methods, then finally physiochemical method. The name itself, it combines both physical and chemical methods. That means use of steam formaldehyde. Steam is physical method, formaldehyde is a chemical method. So it's a physiochemical method of sterilization which takes into account. Now, how is the method is operated? What is the method of operation? That is action of steam as well as that of formaldehyde. In this method, saturated steam, here this method is operating in an autoclave. Okay. So a saturated steam at a pressure of 263 millimeter, which has a temperature of 70 degrees Celsius. The saturated steam is applied. Now the air is removed from the autoclave chamber, and now the saturated steam at subatmospheric pressure is flushed. Then to this formaldehyde is injected with a steam in a series of pulses, each of 5 to 10 minutes. Now the article is then held at this holding temperature. How many time? 1 hour. So after the holding time, switch is off, and the next steps are similar to those in autoclave. Now what are the disadvantages of this method? It may cause what? Condensation of formaldehyde, and therefore induction of large volume of formaldehyde occurs, and this may wet the steam, resulting in loss of latent heat. Then sterilization control, that is using paper strips containing 10 raised to 6. It is 10 raised to 6, okay? 10 raised to 6 spores of Geobacillus thermophilus. So that's all about physical method, chemical method and physiochemical methods of sterilization. In the next class, we can discuss the methods for testing the efficiency of disinfectants. So, thank you all.